Okay.
Hi, everyone. Today I am joined by Dr. Amy Ramsey. My name is Megan Collins, and I'm the research coordinator for CureGRIN. And today we wanted to congratulate Dr. Ramsey on being awarded the Simons Foundation Autism Research Initiative 2021 Genomics of ASD Pathways to Genetic Therapies grant. So congratulations, Amy.
Thank you. We're thrilled. We're thrilled. Thank you.
So a lot of networking and relationship building took place to get this grant submitted and successfully funded. Could you speak to how the grant came together and how CureGRIN played a role in this process?
Absolutely. Yes, it was a process. I think that the first step was that one of the parents and leaders in CureGRIN, Keith MacArthur, went to Boston, talked to a lot of pharmaceutical companies, academics, and biotechs, and to try and find a treatment for his son. And he talked with scientists at Homology Medicine, and they specialize in gene therapy medicines. They were really, I think, moved by Keith's story. And they said, yes, we will help you make the medicine. If you have researchers who want to try medicines in their mouse models, we will make these medicines for them. So he came back very excited about this. And of course, I was interested to try them. I think the next place where CureGRIN really stepped in was that CureGRIN was the one who alerted us to this opportunity. And they knew that we had started the process of getting things going with homology medicine. And they approached me and said, we think that what you're doing is perfect for this grant. Could you please put in a grant? And I was reluctant just because I was doing a lot of other things and I didn't know whether we had a good chance at it or not. But, CureGRIN said we'll help with a lot of the paperwork, a lot of the administrative things. They did everything they could to facilitate it and brought together the different partners in this. So it's a partnership between two academic labs, my lab at U of T, Toronto, and Graham Collingridge's lab at University of Toronto. We have the mice, the different mouse models, that have patient variants. And then homology medicine and Albert Seymour and Jacinthe Gingras at homology medicine. And then a clinician, Dr. Angels Carzola, who is the one who's run the first clinical trial for GRIN. So I think that CureGRIN really brought us all together, did a lot of the administrative things, and that's what made it successful, I think.
We were glad to take part of building the dream team. Can you tell us a little bit about the purpose of this research?
Well, we want to find out whether this particular strategy for gene therapy would work for GRIN1 patients originally, and then GRIN2B patients. So that's the scope of this grant. We're trying one of several different gene therapy approaches that are possible, but this is the one that we think is the most likely to work for the greatest number of patients. The idea behind this is that this gene therapy would work regardless of whether you had a missense variant or whether you had, kind of a nonsense truncation variant. It should, in theory, work for multiple types of patients. So this is why we're trying this strategy of gene therapy.
How exciting. So parents might also want to know what previous work led you to this idea.
Right. So I think that there were two things that we had as knowledge going into this. One of them was that we had already tested the idea of a genetic rescue in our mice. It was not using gene therapy. It was using special tools that are only available for mice. But we knew that if we could fix the gene, then we could fix a lot of the cognitive problems that the mice had. So we knew that it was possible to really improve brain function even in an adult mouse where the brain had already wired together. So that was one piece of information. I think the other piece of information came from labs like Xavi Altafage's lab in Barcelona, where he showed that if you add a variant gene into a healthy neuron, then the variant gene could go and poison the whole subunit. And so our rationale is, well, if we can overexpress the variant and cause a problem to a healthy receptor, maybe we could overexpress healthy subunit, and then we could, overcome the variant. So essentially, instead of poisoning a healthy receptor, we're trying to repair an unhealthy receptor by just adding more of the healthy gene.
That's perfect. Super exciting. So what exactly is gene therapy, and how can gene therapy help grieve patients potentially?
Right. Well, there's so many different approaches that fall within the umbrella of gene therapy. So generally, when we think of gene therapy, we would think of a medicine that somehow involves DNA or RNA. So DNA and RNA are the chemicals that give the instructions to make proteins. And so if you have a change in your DNA, it's going to cause a change in your protein. And that's really the problem. So there's lots of different strategies, but what you need is you need some way to get the DNA or RNA into the cells so that it makes the correct protein. And many times that involves using viruses. And we essentially strip the virus of everything that makes it a virus, and we just use the outside particle because it's really good at getting into cells. So we take out all of the machinery of the virus and we put in the genetic DNA that we want to go into that cell, and then we wrap it up in a virus shell so that it can go into that cell and then deliver what we want to go in there. And that's what we're doing. So there's so many different approaches that it's hard to say what is gene therapy, but essentially what it means is that you're delivering DNA or RNA into a cell to help it overcome whatever genetic problem is there.
Perfect. That's so cool. So your project has 3 aims. Could you talk about those aims and the kinds of experiments that the project's going to involve?
Sure. I mean, I think we can think of the 3 aims as being 3 different representative models. So we have 3 different mice that we're going to study, and we're going to use essentially the same approaches for 3 different mice. So we're going to start with a GRIN1 loss of function variant. And this is one that is coming out of Graham Collingridge's lab. And so he's already studied the variant, and he knows what are the phenotypes of that mouse. He knows what are the problems. There's a lot of patients that also have this particular variant. And so what we will do is try and supply a healthy copy of the GRIN1 DNA to the mouse. And we'll do a different couple of routes of administration. We'll try it just intravenously, and then we'll try it intrathecally, which means into the cerebrospinal fluid with like a spinal injection. And we'll see whether we can deliver enough DNA to change the behavior of the mice to make them healthier. And we also want to make sure that it doesn't cause any problems. So we'll look at the liver, we'll look at the heart, we'll look at the brain, we'll make sure there's no signs of any toxicity. We also want to see how long we can get that benefit. Is it a short benefit, or does it last for a long time? So we'll do that as the first aim. Then the second aim is a different mouse model. It's a gain of function. So this is where the receptors are overactive. And we made that model, that's the model that we have in the Ramsey lab. So, even though you would think, oh, the receptors are too active, we don't want to add more DNA to make more receptor. What we're thinking is that, essentially you've got what you should have is 2 healthy copies, and you have this one bad copy that's causing the problem and it's making the receptor more active. But if we can just add in a bunch of extra healthy copies, that what will happen is that when the receptor is made, it's much more likely to be two healthy copies. So we think it could even work for gain of function variants. And so those are the first two that we'll try. Then the last one that we'll try for the third aim is a GRIN2B loss of function variant. Now, GRIN2B is tricky. As you know, the GRIN2 genes are bigger. And so packaging them in the particular virus that we're using, which is an adeno-associated virus, it's hard because this virus shell, it only will allow so much DNA in there, right? And we're really at the very limits of that constraint with the GRIN2s. So we're going to try it with GRIN2B, but in theory, it would work, it should work the same way for GRIN2A or for GRIN2D or for any of the other GRIN genes. So we'll try to make a virus that can hold the whole GRIN2B DNA, and then we'll deliver that the same way into the GRIN2B mouse. So those are the three aims.
So ambitious. So about how long will this project take, and what does the timeline look like?
Right. So the funding is for 3 years, and that we think that we can accomplish those goals in 3 years. What we may not be able to do is see the mice all the way through to their lifespan. It really depends on how quickly we settle on the correct regimen. And if we get one that works really well right away, then we can do some of those longer term studies. But my understanding from talking with the scientists at Homology Medicine is that this is quite an iterative process, meaning we'll try something, we'll see if it works, and then we'll tweak it. We'll change, the promoter or the route of administration, or, maybe we'll have to add some things to make sure it doesn't go into the liver. And so, that's the goal, is that by the end of the 3 years, we would have the regimen decided, and we would have essentially the specifics decided and have some good information about whether this is viable or not. So it may be that this won't work. It's possible. So we have to recognize that this is not a for sure thing, but we have to try. And I think it has a good chance of working. So I'm optimistic,
As are we. That's so exciting. So what questions from CureGRIN's research roadmap will this project help to answer?
Well, I don't have the questions memorized, but I feel like when we, divided up how we should focus our research efforts, there was a good chunk that was about trying gene therapies. Then there was another good chunk that was about trying small molecules, drugs that could help. And then there was another chunk that was just about understanding the baseline. What are these different variants doing to the receptor, to the body, to the different organ systems? And so obviously it fits into the category of testing out a gene therapy method. But I think we're also going to learn a lot just about how these different variants affect, the receptor and affect the body, because we're still studying these patient variant mice. They're still pretty new, and we're learning a lot. So we'll learn a lot along the way as we test these therapies.
Off the top of my head, I think the questions that we were thinking of from our roadmap that we hope that your project will answer is involving, is a cure possible at any age? What's the best delivery route for gene therapy, and how can we deliver gene therapies for larger genes? So based on the aims, based on your aims, I think we're going to be able to answer a lot of those questions.
I agree. Thank you for reminding me about that. I should have those printed out or like right beside me so that I'm always looking at those. Maybe I will go and do that so that I'm not caught. I'm not caught without the quick answer for that one.
Well, thank you so much. So I think that concludes our questions about the grant itself, but could you also talk about the separate project on stem cell research that's going to be also taking place with Dr. Joe Anderson?
Yeah, now this is really exciting. And again, this is another, you could think of this as a gene therapy in a way, because you are trying to correct something that is wrong at a genetic level. But really, what he has found is that you can just isolate stem cells from the bone marrow, and then you can, so you would take these from the patient, you would correct the problem in those stem cells. So you would fix whatever is the genetic change and make it back to the healthy version. And then you would put those stem cells back in the patient, and you would just put them IV. So you don't have to go back into the bone. You just give them an intravenous injection. Those stem cells will then go and travel throughout the body, including into the brain. And there they can set up shop and start delivering the healthy protein. Now, there's a lot of this that's a lot of hand waving, and because it's not really clear how it is working, but he has observed it working for Angelman syndrome and Tay-Sachs. So it's, there's no reason to think that it couldn't work. So we'll try the same thing with mice, where we will take mouse bone marrow stem cells. We will take the correct version of those stem cells, inject it into our mouse models, and then wait and see, does this lead to, expression of the correct receptor? Do we start seeing improvements in brain function? I am very interested to see whether this works. I have to say, this is a newer approach, but Joe Anderson is at UC Davis. They are set up to make medicine grade reagents there. So I think that in both of these cases, what we have are partnerships with research labs or companies that are really set up to do medicine grade treatments. And this is really different from what I can do in my lab, where I can make something that might be safe for an animal, but it hasn't undergone all of the testing to make sure that it's safe for humans. So yes, we will see about this as well. And CureGRIN is funding that research. They are paying for the reagents in the Anderson lab so that this is possible.
So incredible. We are super excited about everything that you're doing, and we're so grateful for all of the time that you've taken to explain all of the science behind this super exciting project. And congratulations again from CureGRIN.
Thank you. And thank you so much for all of your help, Megan and Keith, and all of you who have really helped us with, giving us the energy to do this, and the support that we need, and, the funding to do some of these early studies that have then led to bigger support like we're getting from Simon's Foundation. Yeah.
Great. Well, thank you so much, Amy.
Bye.
Bye. All right, let me try to stop the recording.